welcome to the next mini lesson in our chapter 3 journey. So this video is going to cover various cell functions that are described in your textbook. They're going to include membrane transport, cell division, and protein synthesis. So those are some big processes that take place within the cell. So we're just going to introduce those and kind of learn a little bit about them. Before we get into the first topic of membrane transport, I wanted to introduce some terms and some concepts that we need to have a good grasp on before we jump into the actual functions of things. So we need to understand the terms solvent, solute, and solution. Okay, so think of I just made some hummingbird food for my hummingbird feeder the other day, so I'll use that as an example. So the solvent in that case is the water. Okay, water is what we call a universal solvent. It's really good at dissolving things. In that case, the solute was the sugar, right? So I'm putting the sugar into the water, and it's going to dissolve. And the solution is the resulting sugar water. I added a little bit of dye, a little bit of pink dye, so that would be another solute. So solutes are the things that are dissolved by the solvent, and when you dissolve solutes in solvents, you get a solution. So the solution was my pink dyed sugar water made up of the solvent water and the solutes, sugar and food coloring. Okay. So the reason why this is important is because as cells, right, so I'm just going to draw a generalized cell with the nucleus, that cytoplasm is mostly water. And the environment around a cell is mostly water. But it's not completely water. So we're going to have things, these are just little squiggles of things that are solutes inside the cell, and things that are solutes outside of the cell. So we're not pure water inside of the cell, and we're not pure water outside of the cell. We're water mixed with some stuff, some sugars and proteins and salts and lipids, and all these kinds of things are all mixed together. And so our cytoplasm is like a solution, and the environment that our cells are bathed in are like a solution, a mix of water and solutes. So when we start looking at membrane transport, we have to understand this relationship between that plasma membrane, right? So our phospholipid bilayer separating the inside world from the outside world. So inside of the cell, it's called intracellular fluid. This is intracellular fluid, and this is extra. So extra like outside. Extracellular. And this is intracellular. Okay? So we are, as cells, all constantly moving things into and out of the cell. So when we talk about membrane transport, we're going to talk about the mechanisms of how solutes can move, or solvents even, watery things can move as well, into and out of the cells. All right, so here on this slide, we have a few different types of membrane transport shown, and I'll draw a couple more illustrations as well. So what we can see is our phospholipid bilayer, kind of the gray layer with the hydrophobic tails kind of dangling towards each other, with the hydrophilic heads pointing towards the extracellular fluid and the intracellular fluid, which is our cytoplasm. And in this first picture, this is our simple diffusion. Okay? So simple diffusion is the idea that substances or solutes can move directly through the plasma membrane. They don't need any helper protein channels or carrier molecules or anything like that. So they just move directly through that phospholipid bilayer. But we have to know something about them. They have to be small in size, and they have to be lipid soluble. The reason for that is because that hydrophobic middle part isn't going to allow things that have charge to them, like water doesn't really move through by simple diffusion. So small lipid soluble substances can go directly through the plasma membrane, and they can go in either direction. But we have to look at some of the other characteristics of the environment to let us know which direction something is actually going to diffuse. So I'm going to make a little drawing down here. I'm going to use some blue X's. So I'm going to draw a whole bunch of X's in this one space of this bottom part of this screen here. So I have a whole bunch of X's clumped together. This is what we call a high concentration. And let's say there's only 2 X's over on that side of the screen. That's a low concentration. Whenever you have things in solution, so like solutes dissolved in solution, you're going to see that the things that are all crowded around each other, this is our high solute concentration, they will begin to move or diffuse towards the area where there's fewer of those things. And they will continue to do that until they reach an equilibrium. So some of these are going to be moving over, and eventually we'll have about the same number of X's on both sides of the screen. Well, if I were going to do it accurately, if the screen were really going to create an equilibrium, I would have X's evenly distributed all over the white space in the screen. I wouldn't have one side that has a higher concentration than the other. So that is diffusion, kind of defined by things move from an area of high concentration to an area of low concentration. You don't need a membrane to do that. They'll do that in a beaker of water, right? Just take a beaker of water, you put a drop of food coloring in there, you'll see a really tight drop of food coloring, but then eventually if you let that beaker sit there on the counter for an hour, the whole beaker will be in uniform color of whatever food coloring you put in there. That's the same idea with diffusion. So what we see with a membrane, all right, so what we see with the membrane is the opportunity to be selective, and the cell can choose what kind of substances it allows to go through. So if I were to delete all my X's and do the same thing, but I'm going to draw a membrane in the middle, and I'm going to have some X's over on one side, a whole bunch of X's over here, and a few X's over here. So we have this kind of the same scenario. We have a whole bunch of X's on one side, so that's our high concentration gradient. We would expect the X's to move through the membrane to reach the other side to try to create an equilibrium. But what if this membrane was impermeable? That's why I'm drawing the red line, impermeable to X's. So no matter how many X's you put on one side of the membrane, they will not be able to diffuse across that membrane because there's a barrier there. For whatever reason, that molecule cannot simply diffuse through the plasma membrane. It's blocked. And so that idea of selective permeability or semi-permeability is the importance of that phospholipid bilayer membrane. So the cell can kind of regulate what are some things that it can take in and what are some things that it can take out. So when we take a look at the next diagram, taking a look at what's called facilitated diffusion, it follows the same principles. We're looking from things going down from a high concentration to a low concentration. So we see those kind of shaded arrows, the reds and the greens, from that concentration gradient. But in this case, these substances are large. They are maybe polar or they have a charge to them. So they're not going to be able to diffuse through the plasma membrane itself, like small lipid soluble things. They're going to be large. So they're kind of labeled lipid insoluble, which means they're dissolvable in water, not fats. And so for those molecules, they have to have a helper. That's the facilitated part. So these purple channels, either a channel or a carrier, channels are just like a tube. The substance can just go right through it, or a carrier kind of changes its shape as it moves through. It's not super duper important at this point, but just know when we talk about facilitated diffusion, what we're talking about is the use of a membrane protein to allow substances to move down their concentration gradient. And both of these processes, facilitated diffusion and simple diffusion, are what we call passive, which means no energy is used. You don't need energy for things to go from high concentration to low concentration. It's just the way it's just what they do. Okay, so that's a passive. So diffusion, osmosis, and filtration are passive processes. Now, diffusion is the movement of any substance from the area of high concentration to low concentration to reach an equilibrium. Osmosis is a specific type of diffusion. It is the diffusion of water across the membrane to reach equilibrium. So let's say we have a cell right? So here's a nice little cell, and it has a concentration gradient of substances inside its cell, or little blue X's. And then outside of it, it's living in a watery environment. And then outside, there's just a few X's. Okay, so we definitely have a higher concentration inside of the cell and a lower concentration outside of the cell. And we can imagine that all the white space is a watery environment. So if we're looking in the frame of reference of water, there's way more water outside of this cell than the inside. So when we take a look at osmosis, this diffusion of water, we're going to use the same rules. Water is going to go from an area of high water concentration to an area of low water concentration. So if we have a whole bunch of water out here, it's going to start moving into the cell, trying to create an equilibrium to dilute the inside of the cell to have that same dilute characteristic that is on the inside. And that's okay in some situations, especially plants are okay to handle that because plants have a cell wall to prevent any damage to the cell. But animal cells, like our cells, cannot protect very much from too much water moving in, and so the cell could pop. So that's one scenario. Let's see what's going to happen if we flip that around. So what if we only have a few solutes inside of the cell and a whole bunch of solutes outside of the cell? I'm probably just only going to do one side for simplicity and time. But let's pretend the whole cell is kind of hanging out in this concentration, a higher concentration gradient. So again, thinking from the perspective of water, water needs to go from high concentration to low. So there's way more water inside of the cell compared to that really salty environment outside of the cell. So the cell would actually lose water. Water will leave the cell trying to go and create an equilibrium and dilute that substances outside of itself. So the cell is going to shrivel up. Okay? So these are different situations that cells can find them in. Either they're completely surrounded by a too dilute of an environment and the cell will gain water, or if they're surrounded by a too concentrated of environment, the cell will lose water. So both of those scenarios are not ideal situations. So what is the ideal situation? So what kind of environment would we want our cells to be in so they don't lose or gain too much water? Well, let's add some solutes inside of the cell. And the best case scenario is we are at equilibrium inside and outside. And we use the word isotonic. Oops. Because iso means the same and tonic means kind of concentration. So if you have the same concentration of water inside and outside of the cell, you are at equilibrium. Water will move equally between both sides of the membrane, keeping the cell at equilibrium with its environment. So that's super important. Okay. I don't think I have a picture of filtration. So filtration, think of pressure, like your coffee filter. So you have this hot water kind of pouring through, you have a membrane that's going to filter the water through and hold some things back. So filtration is driven by pressure, not so much concentration, but it's also passive. Okay. The next type of membrane transport is called active transport. And this means when it's active transport, it needs ATP. Okay, so you have to burn ATP to move things. And if we take a look at the concentration, like an example of a concentration gradient like what we were doing on the last slide, here's all of our X's. All right, so let's get some X's over here. It's not drawing my X's very well. And then let's say we have a few X's down here. Now in a passive or a no energy requirement, we would assume that the X's would go down towards a low concentration. That's exactly what happens in passive transport. But if we are looking at active transport, what actually is happening is substances are moved against their concentration gradients. They are moved towards the area of higher concentration. And that's not a normal process. You need, I mean, it's normal, but it's not free of energy. You have to put an input of energy to move things back towards an area where there's a whole bunch of stuff already there. So I have this illustration up here towards the top is we have a high concentration of hydrogen ions outside of the cell, and only one hydrogen ion on the inside of the cell. And so we use the power of ATP to move that hydrogen to another side of the membrane. And that is really important for cell processes to work. This one down here is called the sodium potassium pump. It is a very common membrane transport protein that uses the power of ATP to pump. So the green and the yellow arrows are representing the concentration gradients. So sodium usually would like to flow in, potassium usually would like to flow out. And so we are using, let me get the yellow, ATP to move sodium out and move oops, that was supposed to be green, and move potassium in. Okay, so we are moving these substances against their concentration gradients, and we require ATP to do that. So in the job of the sodium potassium pump, one ATP molecule can move 3 sodiums out and 2 potassiums in. And this is actually going to maintain homeostasis for cells that need that difference and that separation of sodium ions and potassium ions. You're going to be learning a little bit more about that in the muscle chapter and the nervous system chapter when we learn about how muscles and neurons work. All right, this last part of membrane transport is going to be our active transport. So these are big scale active transports. We use energy for these, but they're called exocytosis, endocytosis, and pinocytosis. So exo, meaning outside, and cytosis means of the cell. So exocytosis is when a cell is moving things and releasing it outside of the cell, and it's a big bulk process. Endo, meaning inside or into, and so when we are taking in substances, ingesting substances, this is called endocytosis. And then pino is very much like endocytosis, but it's more for liquids only. Sometimes pinocytosis is called cell drinking, and endocytosis is cell eating, or we also had the other word phagocytosis is a type of endocytosis. So exo is releasing, endo is taking in, and pino is drinking. All right, let's see. Okay, I think what I'm going to do is I'm going to split the cell function into smaller videos. I'm already at almost 18 minutes, so I'm going to stop here and I'll do the cell division in its own video and the protein synthesis in its own video. All right, see you for those.